speaker, Dirk Trauner. Dirk is a chemist, and I think he's best described as a chemist with many interests. And he's probably one of the first person who really reused and popularized the term photopharmacology. And so he will give us an overview and introduce us to the use of light to control at the molecular level molecules, and then we will see what effects this may have. So he is the person kind of representing today the field of chemistry, medicine, and applications in molecular biology of light on there. With this, I would like to give the word to you, Dirk, and we are happy that you're here.
Thanks, Burkhard, for this lovely introduction. Let me share my screen. And thanks for putting this together. This is actually quite exciting. This is the first time that I'm participating in something like this. Can you see my presentation? Let me start it on photopharmacology.
Excellent, perfect.
So as Burkhard mentioned, photopharmacology is an attempt to control biological function with light responsive molecules with, as you will see, little switchable molecule fascination with light and molecules. Photochemistry is an absolutely amazing topic, and as chemists we really love working on this. Just to illustrate how amazing light is, daylight that is, this is a picture of the Andromeda galaxy. Right? The Andromeda galaxy is the furthest object that you can see with your unaided eye. It's about 2.5 million light years away, which means that a photon that emanates from the center of the Andromeda galaxy travels for 2.5 million years, and then through the atmosphere and through your tear film and through your cornea and through your lens and through your vitreous until it hits the retina, this light responsive tissue, where the molecular, the chemistry is happening. And that happens at a ridiculously low intensity. This happens at 10 to the 5 photons per square centimeter per second. Just to illustrate this, if you go skiing the next day after you've seen the Andromeda galaxy at night, you have 10 to the 17 photons per square centimeter per second on a glacier, on an ice field, which means that the human vision spans a dynamic range of 12 orders of magnitude. And there's really nothing else in physiology or in biology, for that matter, that does this. And that's one of the reasons why we are so fascinated with it. But of course, we are also fascinated by the biology and by the chemistry that happens in your retina. This is a very simplified diagram of your retina. Let me actually move away these pictures of yours. Here we go. So I can read my own slides. It's a diagram of the retina that shows that the retina essentially consists of 3 layers. They are the rods and cones, the so-called photoreceptor cells, which express, as we will see in a moment, a type of protein called rhodopsin or opsins that does the actual photochemistry. And then these rods and cones talk to a layer of interstitial cells, bipolar cells, amacrine cells, whatever they're called. And finally, this information is conveyed to the retinal ganglion cells, GC, which project the nervus opticus into your brain, where the information then is finally processed. But I have to say that a lot of the processing is happening already at the level of the retina. The retina really is a part of the brain that is put forth, and where much of the processing already happens. So what happens actually on a molecular level? What happens on a molecular level is shown in the next slide. Here it is. What happens is that a small organic molecule, a sort of fatty lipophilic molecule called retinal, which is bound to the protein, undergoes an isomerization. There is a form which is spent. This is the so-called 11-cis form. And upon absorption of a photon, this double bond here in the middle isomerizes, goes from cis to trans by rotating literally. And this rotation is actually felt by a protein envelope that encumbers, that sort of embraces this molecule. This protein envelope is shown here in a little movie, so you have to imagine that retina, the organic molecule, sticks in between here. This is my surface rendering. And this protein is not just a protein that circulates around in the cell, it's actually bound to the membrane. It's a transmembrane protein. And this transmembrane protein, as shown here in all its glory, is embedded in the lipid membrane of your photoreceptor cells. And when this action, this movement happens, ultimately through a very complicated process, this results in a change of the transmembrane potential and in a change of the release of a neurotransmitter called glutamate. And then many other things happen. It's a really complex series of events, but ultimately the retinal ganglion cells fire action potentials, and that is what you perceive as light at this extremely low intensities as well. So it turns out that in certain types of blindness, the photoreceptor cells are gone, are degraded. This happens, for instance, in macular degeneration. This happens in late stage, in retinitis pigmentosa, and in late stage macular degeneration. You lose the rods and cones. The cones, by the way, are responsible for daylight vision, so this is more relevant to this meeting. The rods are responsible for night vision. We have evolved two types of visual perception to, again, span this enormous dynamic range. And both of them are gone. They are, for various reasons, degrading, which means that the retinal ganglion cells at the bottom here don't receive any input anymore. And therefore, you turn blind. However, if you could hijack some of these other proteins, shown here on the right side, there's a whole laundry list of other proteins that are expressed in the bipolar cells and expressed in the retinal ganglion cells, and also these other inhibitory cells, amacrine cells, horizontal cells. If you could turn these proteins into photoreceptors by endowing them with an artificial photo switch, you'll see in a moment which one it is, with a chemical that is light responsive, perhaps this sort of information transfer could be sort of hijacked, could be bypassed, if you will, the need to input from the photoreceptor cells. And perhaps you would have some neuronal output here, some firing along the nervus opticus that would be perceived as vision. And this was one of the goals of our research program, which we started some 17, 18 years ago now in Berkeley, teamed up with a fantastic team of neuroscientists to make this happen. So we are aiming at artificial vision, which uses artificial synthetic molecules that are also light responsive, like retinal, undergo the same form of change, of bending, of isomerization, as we call it, to restore visual responses. And just saying that this has mushroomed in a very broad sort of effort, going through all these different proteins which we have endowed with a photo switch, but we've become quite adept at doing this, and we call this, as Burkhard mentioned, photopharmacology. All right, here is the little photo switch that could. This is a molecule that is very sort of familiar to organic chemists. Everybody who went through a sort of introductory organic chemistry lab probably had to work with this at some point. It's an azobenzene molecule. It's artificial. It does not really occur in nature. This is something that humans have invented in the 1820s, and then in the 1830s, in the 1930s rather, it was established that this molecule undergoes an isomerization upon irradiation. So when you take the parent molecule, azobenzene itself, you have to use UVA light to bend it to the cis form, from the long elongated trans form to the bent cis form. And then with 500 nanometers with green light, you can bend it back, or you can just let it sit, and it will isomerize back. But that's just a sort of parent molecule. We have over the years created many other molecules that have slightly different photophysical and photochemical properties. For instance, this molecule DNAC here, this molecule is an azobenzene that can be bent with blue light. For 72 nanometers is blue light. And then once it is bent, it actually falls back very quickly, within milliseconds. As soon as the light is gone, thermally it isomerizes back. It turns out that this molecule is a blocker of an ion channel, of the famous potassium channel that is all important for neuronal activity in the trans form. It is actually a photo-switchable version of lidocaine. That's something you're all familiar with, I assume, because when you go to a dentist, you get an injection of lidocaine to numb your nerves. And this molecule in the stretched long form indeed numbs the nerve because it blocks potassium and also sodium channels. Whereas if you bend it to the cis form, it does not block, and therefore allows neuronal activity to happen. And this very, very simple principle was used to make a blind retina photosensitive again. Let's take a look at this. So we are applying this molecule. This molecule, by the way, mostly targets retinal ganglion cells. So these are the cells that spike, that fire these famous action potentials. And they do now in a light-dependent way. In order to investigate this, we use some beautiful electrophysiology, which we can irradiate with blue LEDs. So thanks to all of those here who have been involved in developing and optimizing LEDs. They're just awesome. We absolutely love them. There is a Nobel Prize for a reason for this. And here is the situation of a blind retina before we add the molecule. What you see here on top are individual traces that sort of measure the electrical activity around these retinal ganglion cells. So these are so-called extracellular field potentials. Doesn't matter what it is. It gives you an idea of how spiky, how active they are, these individual traces. Then you sum these traces on the bottom and you get this noise, right? It turns out that it doesn't matter whether you shine white light on it or darkness, no light, white light, no light, white light, no light. There's absolutely no response, and that is expected because this is a blind retina. It's a blind mouse retina, by the way. And when we add now our molecule, very simply, we sort of add the molecule to this tissue, we see this dramatic change in neuronal activity upon irradiation. So when you irradiate, you see a large increase in spiking frequency. This sort of is an idea of firing rate, how quick these nerve cells fire, whereas in the dark, it's much less. It's much more, much less, much more, much less. This is a somewhat older slide from 2014, but it was the first demonstration that this works. And in the meantime, we've applied this to more elaborate models of blindness. Stay tuned. It's not in humans yet. We have gone through the safety studies and we have demonstrated efficacy in higher animals, but in order to demonstrate this in humans, we have to go through a few more steps of drug development. In any case, if this works, it would be a rather simple way to restore vision that, in my opinion, could be competitive with optogenetic methods. You're probably aware, you might be aware that there are some gene therapy approaches to do this, and there are also some devices which you implant into the eye to electrically stimulate the retina. We do this with molecules that we can chemically stimulate. Let me move on to a second topic where photopharmacology, I think, can move fast in terms of clinical translation, and that is cancer. Right? It turns out that cancer, as you all know, is an uncontrolled growth of cells, which, of course, require the cytoskeleton to grow and to divide. So this network of fibers of actin, tubulin, intermediary filaments, you name it, that control the shape of a cell, but also are crucially important for cell division, for pulling the chromosomes apart, for instance. This is done through microtubules. And here's a beautiful rendering of a cell where the actin cytoskeleton is rendered in red, whereas the two microtubule cytoskeleton is rendered in green. And it turns out that these microtubules have fascinating dynamic instability. These are the cables that pull in a cell and that push to a certain extent. And these are tubes that are made up of proteins that come together and fall apart. This is a highly dynamic process. This is the so-called dynamic instability of tubulin. And we can control this dynamic instability with a photo-switchable version of a very famous poison. So it turns out that colchicine, that comes from the autumn crocus, from Colchicum autumnale, it disrupts these microtubule dynamics. Here's the structure of colchicine. It turns out the colchicine has a little cousin known as compretastatin A4, which has a cis double bond. It has this double bond where two things are on the same side, called a cis double bond, instead of this 7-membered ring. But other than that, it has this rim that looks somewhat similar. And indeed, it mimics colchicine, but only in the cis form. And we could not resist from putting an NN double bond in the middle to make it an azobenzene. And now the azobenzene is presumably active in the cis form, which you can achieve by irradiating with violet light with 400 nanometers, whereas if it falls back to the trans form, it becomes inactive. And this was developed together with a postdoc of mine, with Oliver Thorn-Seshold, and was demonstrated to actually work beautifully. So what you see here is a cell where the microtubule dynamics has been visualized. Let's start the movie. This sort of hectic buzzing activity is the microtubule network as it forms and as it disassembles and forms and disassembles. And when you don't add this molecule, which I just showed you, this azobenzene, then nothing happens. The cell just sits there. The microtubules form and disassemble as they should. This is called the dynamic instability. However, if you add the molecule, nothing in green, and then stop. And it goes again, and stop. And it goes again. And this translates to the optical control, to the light control of cell proliferation. What you see here are two cells in an embryo, in a C. elegans, in a warm embryo. These cells are synchronized, as one says. They would go cell division at the same rate. The chromosomes are lined up in both cases, but the one on the left side, we irradiate, and we prevent the chromosomes from being pulled apart because these cables are not functional. Whereas on the right side, they do get pulled apart, and that, of course, ultimately translates to the optical control of cell division, which is exactly what you want to do in cancer. You want to prevent cell division, uncontrolled cell division, but you want to do this with the precision, with the ultimate precision that light affords. Light is unbeatable, I would even say, in its ability to temporarily and spatially control action, right? And that remotely as well. So our dream, our grand vision is, let's go back, our grand vision is that we make molecules that are inactive in the dark. They're not toxic, like other microtubule disruptors. The most famous one is Taxol, right? If you ever talked with somebody who underwent chemotherapy and had to take Taxol, I can tell you this is horrible, this is an ordeal, because Taxol acts everywhere, right? And there is little sort of selectivity other than that cancer cells divide much faster than other cells. But the hair follicle cells, for instance, that divide fast are affected, and this is where you lose your hair when you're on cancer chemotherapy with Taxol. And these are exactly the side effects we try to avoid by making molecules, pharmaca, drugs that you can turn on and off with light, like the one you've seen before. So the motto is apply globally and activate locally. I think I have 5 more minutes. Am I correct? Hello?
Yes, you're good in time. So 5 more minutes is good. Okay.
5 more minutes, and I will top this off with something that is brand new that we have just put on the web and will soon publish. This concerns a class of molecules that mark proteins for destruction. When we would like to control these molecules with light. These molecules are called PROTACs, proteolysis targeting chimeras, and they're currently all the rage in pharmacology. This is one of the hottest fronts in cancer therapy in pharmacology. These are molecules, pretty big molecules like the one shown here, that grab a protein and then make sure that this protein gets flagged for destruction. Proteins in a cell get destroyed in an orderly fashion. They don't just fall apart after time, but they actually get destroyed through the so-called proteasome system, ubiquitination, just throwing out a few buzzwords. There were Nobel Prizes for this, of course, as well. So the life cycle of a protein is not only consisting of how it's made on the ribosome, but also how it is destroyed. And this flagging a protein for destruction with a synthetic molecule is really exciting because this molecule can function as a catalyst. It can turn over, and therefore you can have very low concentrations of a molecule that promotes this flagging. And these molecules are called PROTACs. And here's one, which, by the way, on the left side has the structure of thalidomide. This is nothing else than the infamous Contagan, originally marketed as a sleeping pill, sadly, but has made a comeback as an anti-cancer compound because the mechanism was figured out, what really happened there. And this is now a multi-billion-dollar drug known as lenalidomide or thalidomide, which has this makeup here. And on the other side, you have a ligand for whatever, which you want to destroy. And we could not resist putting in a photo switch, so we put in our azobenzene, shown here in the cis form, replacing this part, so to speak. And the dream was that this molecule is inactive in the dark, and the azobenzene is trans. But if you summarize it to the cis form, this is with UVA light or with violet, deep violet light, then it becomes active. And indeed, this is true. These are so-called Western blots. Let's look at one lane here. This is just what happens to a protein, these little mice track ovals here. This is just a visualization of a protein. A protein called BRD4, doesn't matter what it is, but it's critically involved in reading the genome. And you see that this protein is not degraded, is not destroyed in the dark in the presence of our compound in different concentrations, 30 micromolar to 100 nanomolar, 30 nanomolar, nothing happens, right? But as soon as we turn the light on, this protein gets destroyed at a concentration of 1 micromolar, 300 nanomolar, it's gone. And this has interesting consequences for those into it, it actually leads to a control of CMYC, which is a very important target for anti-cancer drugs. So we're quite excited about this, and we can do this pretty quickly. So it turns out, whoops, what happened? As the shared window is closed, let me go back. I'm sorry, I think I just lost my presentation. Let's do this again. So this degradation is happening fairly quickly. Let me share it again. I'm not sharing at the moment. Okay, let's go down. I'm almost at the end. This is happening quickly within a few. Oh, I'm sorry. I don't know what's happening here. I'm hitting a. So, in other words, we can control this process, and I just want to throw out that if you want to hear more about this whole, first of all, I want to show you the hero behind this, my graduate student, Martin Reinders, and the postdoc by Matsura, who developed this. These, by the way, are these sort of cell disco systems with which we analyze protein levels. So it looks also very good. Has this beautiful LED look, this rainbow look, the lab. And I want to conclude by saying that photo switches, if you wonder whether this can be clinically used, are indeed clinically relevant. So light and medicine is a huge topic. There's such a thing called photodynamic therapy. We are trying to elevate photodynamic therapy to a new level of sophistication. And if you wonder whether irradiation and babies are a good idea, I want to remind you that in order to cure neonatal jaundice, where you accumulate bilirubin, a degradation product of blood cells in large quantities, and then babies turn yellow. In order to cure this, you actually irradiate this molecule with blue light. You undergo you elicit a photoisomerization. The molecule can be becomes soluble and then can be excreted. And this is done in neonatal wards by wrapping babies with blankets of LEDs. So if it's good for babies, it must be good for us. And therefore, I'm quite optimistic that photo switches will ultimately find a more expanded role in medicine. If you want to hear more about it, by the way, I would like to invite you to a conference which I'm organizing in New York City, Photopharmacology 3, taking place May 28, 29, 2020. And soon you will hear more about it. The website will be set live. For now, I would like to thank you again, Burkhard, for inviting me. And if there are questions, I'd be happy to answer them.
Perfect. Thank you very much, Dirk. There was a great overview and introduction to the field, and I think it was very exciting. Our audience is a little shy. So far, they don't have placed any questions. So just to remind everybody listening, you can use either the chat to put a question or the question and answer. Just put the question there. I can read it and put it on and we'll ask it then to Dirk on there.
Am I still sharing the screen, by the way?
No, it's fine. It's good.
Okay, I'm not sharing the screen anymore. Okay.
No, it's good. It's all good. No. Let me start off because of course I know the field a little bit. What is in your opinion the current need on there? We see the molecular switches are already fine, so chemistry is good. There is still room for improvement.
I would say better switches are absolutely needed. Heterocyclic azobenzenes, okay. Right? So I would say that better switches that allow for deeper penetration of light, switches on the red end of the spectrum that are still thermally stable are very important. So more chemistry, better chemistry is always needed, right? That's always possible.
Right. Yeah. Yeah, I fully agree. I think this is something we definitely need to work on. You mentioned the fantastic possibilities in pharmacology applications on there. But I remember talking also with Ben Feringa, who is also has some projects in the area on there. And so his impression from the feedback from pharmaceutical industry was not so positive. So what is your opinion or your impression on this? And I'm also not, are there currently drugs in development in pharmaceutical industry that use these principles to control activity by light?
Yes and no. I mean, we have a startup on this, and if you can call this industry, it's not big pharma at this stage. So the larger pharmaceutical companies have not picked this up yet, because this is very risky at this stage, and it's very, how shall I say, exploratory. And industry in general, pharmaceutical industry, does not like photoactive molecules because they associate photoactivity with phototoxicity, which usually means production of single oxygen. And that is exactly what these photo switches don't do, right? They do not produce single oxygen because they are not photosensitizers for oxygen. They are photopharmaceuticals. They isomerize instead of giving the excess energy to oxygen and making single oxygen. So I think it will require a little bit more of education, right? It turns out that I actually have some ties now with local industry here in New York, especially in the product context. As I said, this is currently a big deal in industry, and they are very primed towards thinking about how to make this even more effective. There's some concerns that these products are too toxic because they turn over, they function like catalysts. So in other words, if you could switch off the toxicity at will, and not just wait for clearance in the metabolism, this could be useful. Right. As every brand new concept, it will take some time until this transpires. But we keep pushing. I think I have given enough talks now in industry to lay out the principles. There is some concern about how to deliver light, and I see there is a question, I think, in this.
Yeah, questions are now upcoming on there. Let me phrase the question so that everybody can see this. So there's a question about future applications for neurodegeneration diseases like Alzheimer. Do you see something in this direction developing or possible?
This would be wonderful if we could dissolve plaques with photo switches. I think there is a possibility. Especially, and I just wanted to mention this, since light delivery methodologies are becoming very, very sophisticated now, to the extent that you can think about drilling a hole in the skull and lowering an LED. There are devices by John Rogers, for instance, that are about 8 millimeters in diameter, 1 millimeter thick now, that can, and even smaller ones are possible. So there are 50 micron LEDs that you can implant. This was actually one that is wirelessly powered I was referring to. So I think we won't shy away in the future of interfacing humans with electronics, and if these electronics emit light, all the better. Right? I don't think I have to explain to my colleagues here that this is a very hot and very interesting field. As I said, we want to elevate photodynamic therapy to a new level. So the light is used in medicine. By the way, photomedicine is old as medicine. The Egyptians have used it, right? Psoralen was used by the Egyptians, and the third Nobel Prize in medicine went to Niels Finsen for photodynamic therapy, for phototherapy, right, of lupus vulgaris. Now obsolete because there are antibiotics. But so the idea of using light for therapy is, to a certain extent, implemented in medicine. We just have to become more sophisticated with chemistry. In photodynamic therapy right now, you only make single oxygen, you burn everything around. You cauterize around and you maybe elicit the immune system a little bit. We think we can get better than this, in part because the ability and the methods to deliver light are becoming really sophisticated. And of course, we're also in lockstep here with optogenetics, because in optogenetics, you don't only have to deliver a gene. There are a lot of people who think about this in human therapy, but you also have to deliver light. And
right, yeah, there was also, maybe you can address this quickly because we're running out of time a little bit. How do we get the molecules, the photo-switchable and then activatable molecules, at the right place in a body? That was one question.
You can target them with nanobodies, with antibodies. So we have published on photo-switch antibody conjugates to make them very, very selective. Or you have good pharmacology, right? Yeah. So again, the idea in principle could be that they are everywhere, that they don't have to be targeted to specific cells, but they're only active where you irradiate them. Right. And then they're harmless, ideally, everywhere else. But we can also add additional targeting motifs, such as antibodies and nanobodies.
Right, yeah. How far are we away from human application in cancer treatment?
Cancer, it's very hard to say, because we've just started with mice. And
okay, so
everybody can cure cancer in mice. So this is the sad truth. And it could happen very quickly. We're all waiting for the first clinical data on the products themselves. So this happens this year. If they are clinically relevant, if this becomes a drug, Craig Cruz will get a Nobel Prize, right, for this new principle. I think we will be able to move a little bit faster. For the cytoskeleton targeting molecules, you should ask Oliver Thorn-Seshold, who has been at the forefront of putting this into animals. It's hard to tell. It does work in animals, but again, we're at the mouse level, and the mouse level is the lowest of all. And we have all learned that projecting from mouse data to human data in cancer is delusional.
Right. Yeah. There was one specific question, and that's the last question, on the mechanism of the switching of the PHOTAC. Why is this? This is a cis-on ligand. And how do you realize that? How do you do
I wish I could answer this question. I hope this is not one of my reviewers because I got this question. It's an extremely interesting question. It's a very important question. If the compound had been active in the dark, it would have been much less useful unless you make them bistable. But as Eric Carrera and Craig Cruz did recently, by the way, but we do not understand at this stage how they work exactly. And in all truth, nobody in the PROTAC world understands how exactly these molecules work, and where the slightest change in a molecule, making the linker just a little bit shorter, completely destroys activity. But it's exactly what we do. We change the linker length and orientation with our photo switch, and therefore chances were high that this does work. But stay tuned. We only have one paper out at this stage. I'm sure that I'll be in this business for 10 years at least.
Perfect. Thank you very much, Dirk, for giving the insight on the photopharmacology. It was really great. Just to every